We're going to talk about mitosis now. We finished with cell cycle, G1S, G2, MC, and how the cell goes through a life cycle of starting small and growing to full size. That's G1, first growth. As it becomes full size, it kind of stays in that position for a while and just does its job. Whatever tissue it's a part of, that cell is usually just doing a job in the body. After G1, now you're starting to prepare to divide. And so the S phase, synthesis phase, is where we copy the DNA. G2 is called second growth. It's where we make extra organelles as the cell prepares to divide so that each new cell can get the organelles it needs. And then the M phase stands for mitosis. That's what we're going to talk about now. C, cytokinesis, where the cell actually breaks into two cells. So first of all, G1S and G2, when we begin the process, the cell looks like a normal cell. You have the cell membrane, you have the nucleus. Inside the nucleus, you might see some blots of DNA, but you can't actually see chromosomes because the cell's not going through mitosis. Therefore, the chromosomes have not condensed or formed yet. You can't see them, okay? So this is G1S and G2 kind of collectively. Your book calls that interphase, where it's basically in the time of life where it's not dividing. Over here, now we're going to start the mitosis process. Remember, mitosis is all about creating genetically identical diploid cells. So in this first cell, we call it prophase, where we're beginning the process. You see in here 8 chromosomes. What is breaking down is the nuclear membrane. So over here, we had a clear nucleus, but now the membrane around the nucleus is breaking down and starting to dissolve. That's going to make room for these chromosomes to organize and split. The very next step, the nucleus is gone, and the chromosomes can kind of line up down the middle of the cell. So we call that metaphase. So the chromosomes are all lined up. At this point, there'd be parts of the cytoskeleton that would grab each chromosome, okay, and set it up so the chromosomes could then split and move opposite ways, okay? So what's happening here, if this is an animal cell, by the way, is that these chromosomes will be pulled apart by centrioles. Those are those organelles that help animal cells divide faster. And they would be found back here pulling these guys apart. Remember, this X really represents, okay, that X represents 2 copies of the same DNA. Back in the S phase, we copied the DNA. Because before S phase, we only had one kind of chromosome. We had one copy of the DNA. We made a second copy. So if that's a gene for blue eyes, then right there, that's a gene for blue eyes. If that's a gene for a disease like, say, cystic fibrosis, well, that's the gene for the same exact disease, cystic fibrosis. We copied a genetically identical copy of that strand of DNA to make a second strand. So that X really is 2 copies of the same DNA. Now they're going to split apart. And so we call this anaphase, where the chromosomes get pulled apart from the middle to opposite ways. And so they're getting grabbed and pulled. So here we see them lined up, here we see them separating. When the chromosomes are far enough apart for the cell to start dividing, here we call this telophase, okay? Telophase is where the chromosomes are separated. Telo, remember, is Latin for distant. So you have a telephone if you want to talk to somebody far away. A telescope if you want to see something far away. Where telophase is where the chromosomes are far away. They're as far away from each other as they can get. And now you start seeing the cell dividing here. That would be the beginning of cytokinesis, okay? Once the cell is fully divided, now we notice we go back, we still have 8 chromosomes. Remember, we started with 8, we end with 8. But now, instead of having duplicated chromosomes, we have just a single leg here. We just have unduplicated chromosomes. Again, because now we're back in that G1 phase where we're going to grow to full size. And that single unreplicated chromosome is going to form a second copy when this cell goes through S phase. And so again, it's this whole cycle of life. G1S, G2M, C. C is, now we divide it into two cells, and each of these two cells starts over in G1, then to S, where we duplicate the DNA, then to G2, then to M, where it would go through this process and divide again. And this is how cells go about making, the cells stay diploid, okay? Because I start with 8 chromosomes, I end with 8. I still have mom and dad's copies inside of this cell, and those same copies get sent on to this cell. So I start diploid, I end diploid. And I'm creating genetically identical cells because I'm just taking my 2 identical copies, splitting them, and making sure that one copy goes to one cell and one goes to the other cell. That's mitosis. Now I want to remind you of meiosis. Okay, I want to remind you of that process where we, actually I'm just going to move over to this table. All right, where we go through the process of creating gametes, sperm and egg cells. And again, here we're not staying diploid. We're starting with a diploid cell with our 8 chromosomes. But the biggest difference is in mitosis, those chromosomes lined up individually, 8 down in a straight line. In meiosis, they line up with what we call their homologous partner. And so here, the 2 partners, they line up next to each other. What these homologous partners represent is they represent mom's chromosome and dad's chromosome. Same chromosome, but different alleles, because mom and dad are going to have different versions of certain genes. And so, for instance, you know, if I were to blow up these 2 chromosomes, they might look like this. And this might be the eye color gene. Well, that's going to be eye color gene. But let's say dad had brown eyes and mom had blue eyes. These chromosomes are homologous. They're similar, but they're not identical because they contain different alleles, one from mom and one from dad. So in meiosis, those homologous chromosomes line up next to each other, because what we want to ensure is that our sperm and egg cells get one copy of the eye color gene. It might be brown, it might be blue, okay? That's the whole idea of genetics and Punnett squares. If I start big T, little t, those split, and the big T goes to one cell, and the little t goes to a separate cell, okay? And so that's what's happening here. Imagine this is, you know, big T and this is little t. Well, they split, they go to opposite sides of the cell, and the big T goes to one sperm or egg, and the little t goes to the other sperm or egg. Well, which one ends up fertilizing, you know, and making the baby? Well, that's why we create the Punnett square, because it's a 50-50 chance. If this cell that contains a big T gets passed on, well, then the baby got the big T from dad. If this cell that has the little t gets passed on, well, then that goes into here. And then we look at what the other parent gave. Point is I just wanted to remind you about how mitosis is creating genetically identical cells that stay diploid, but meiosis is creating haploid cells, sperm or egg, with just half the chromosome number. And the process, of course, looks a little different because the homologous chromosomes line up together in metaphase. Then they split, they go to opposite sides. So when this cell divides, I started with 8 chromosomes, but now I only have 4. So now we're haploid, okay? Now this process is going to go through another division because I still have my second copy. And so this basically goes on to look a lot like mitosis, but with half the number of chromosomes. So the X's line up, the X's split into the individual chromatids, they go to opposite sides. So I end up with four gametes from one, you know, what we call germ cell, basically the cell that's going to divide into those gametes. So one germ cell will then eventually produce 4 gametes through 2 sets of divisions. Each of these guys would get 1, 2, 3, 4 chromosomes. Here, 1, 2, 3, 4 chromosomes. And then here, 1, 2, 3, 4, 1, 2, 3, 4. Okay? So again, meiosis versus mitosis. Okay.